Thank you, Stephen, for the kind introduction. Let me share my slide. Uh, Share screen. Can everyone see my screen? Okay, great. Okay, good afternoon. So actually, I'm going to present about using deep learning neural networks to predict splice site in DNA sequences. So my name is Kuan-Hao. I'm a third-year computer science PhD candidate. So today I will discuss how to predict splice site in DNA sequences with sequence-to-sequence model. And first, I'll generally talk about the sequence model, and then describe some of the work I've been working on and have been done, and also share the perspective of the future of applying transformer-based sequence models. And with that, let's get started. For the purpose of this talk, when I mention sequence model, we'll think of it as a black box that maps one sequence to another. These models is composed of primitive layers where each layer simply takes an input sequence into a model and return the sequence of the same shape. But this can be easily converted to higher dimensional features. And many sequence models have been developed to satisfy the interface, particularly in the context of deep learning. These include many classical deep learning models such as recurrent neural networks or RNNs, and convolutional neural networks or CNNs, as well as more modern model architectures such as transformer and neural ODEs. And all of these models kind of satisfy the same interface. They map a sequence to a sequence, and then you can incorporate any of these into a deep learning model fairly easily by just using a standard architecture, where you can first use the normalization layer, input it into a sequence model, and add a linear or nonlinear activation function, as well as residual connections. And for this talk, I'm mainly going to focusing on the approach of CNNs, which have been proven very successful in capturing long-range dependencies and interactions in DNAs. I will also spend some time discussing some of the recent work that people are excited about of transformer models. And these generic deep sequence models can be used to solve many different types of problems with many types of sequencing data, from modalities such as text to more general time series data such as audio, images, and videos. Furthermore, in the application to genomics, it has been applied to protein sequences, and this is a figure from Meta's ESM-Fold. And also DNA sequences, and this is a collaboration with DeepMind and Calicos model, Informer model. And DNA sequence is the main topic that I'll cover today. So putting them on a spectrum from discrete to continuous, I'm going to draw a very rough distinctions between different types of sequence data. Now, many modern sequence models have been focusing on data such as video, audio, and more generic time series data. And what's common to all of these is that there's an underlying notion of time from which the data is is sampled from. And in contrast to continuous data, what I'm focusing on today is roughly more discrete, and I'm going to roughly call them discrete sequence because input comes in the form of discrete tokens, for example, like DNA nucleotides. So CNNs and DNA sequence. What are the strengths of applying CNNs to DNA sequences? I'm using one slide to demonstrate the impact of applying CNNs to learn the underlying context of DNA sequences for predicting various of tasks, and then I put them into a timeline. So the earliest successful attempt starts in 2015. DeepSeq and DeepBind were the earliest works that introduced deep learning-based models to DNA sequences. These models predict epigenetic marks directly from DNA sequences with CNN sequence model. And CNN models has also been applied to learn patterns in the DNA sequences with Hi-C labeled data to predict 3D genome organization from the DNA sequences. And furthermore, the more widely used of CNNs is in predicting gene expression, following the success of early sequence models in predicting chromatin profile prediction. CNN-based models has also aimed to go beyond epigenetics and directly predict gene expression from sequence. Models include Basenji, developed by Calico, Expecto from the Cherokee group at Princeton, and Informer by DeepMind and Calico, and so on. And finally, CNN-based models have also been proven very successful in splice site prediction, which is the main topic I am focusing on today. And the most representative one is Splice AI, developed by Illumina. Okay, so that's a lot of model. Before I dive into my work, I want to share a quote from Dr. George Box. All models are wrong, but some are useful. So far in my PhD, I've been mainly working on genome annotation. And a crucial component of genome annotation is the identification of splice site. Splice site marks the boundaries between exons and introns, and recognizing splice site is important because it helps us to more accurately assemble transcript and also decide the gene structure. And I'm developing deep learning sequence-based models to understand the underlying patterns in the DNAs to predict splice site. I will talk about two tools I'm currently working on, SPLAN and Splice AI Toolkit. So here's an example. Given a pre-mRNA sequence, in this example, there are two splice junctions. Spliceosomes splice out two introns and produce the mature RNA. So now let's try to convert this biology problem into a machine learning problem. Given a pre-mRNA sequence, a string composed a list of A, C, G, T alphabet, and this is Rx. And for each position, we can label the donor site as 2, acceptor site as 1, and neither as 0, and this is Ry. So now the problem is transformed into a sequence-to-sequence model, and we can use machine learning method to try to solve it. And the current state-of-the-art tool is called Splice AI, which was published in Cell back in 2019. So the way to use Splice AI is you select a gene sequence of random length, and you append 5K and 5K flanking sequence on each side. You do one-hot encoding, feeding to the model, and the model will tell you for each position if it's a donor, acceptor, or neither. So we have XY pair, we can train the model, but you may have the question that how are we going to do the batch training and batch prediction, and also how to do the stochastic gradient descent because the input sequence are in different varies of length. So the approach, yes. Yeah, that's a good question. Widely, people just simply do one-hot encoding because ACGT, they treat it as like equal contribution. And also AND, they just use 0, 0, 0. But you can definitely use like calculate some probability and give weight to other like specific label. Yeah. Okay, so the approach that Splice AI adopted is called sliding window trunking. So given a gene sequence, we can label X and Y. So now we select a window size in Splice AI's case as 5K, and we put flanking sequences on each side, also 5K on each side. And we slice through the gene sequences, and now XY, a gene sequence is converted into an XY matrix, where X is in the shape of L over W, taking the upper bound times the flanking sequence plus the window size. So now zoom out to the full genome annotation. We focus on the protein sequences, and we gather roughly around 19,000 to 20,000 protein coding genes. Using the same sliding trunking approach, we can convert them into XY pairs. And now we can use this approach to do the stochastic training on Splice AI. So Splice AI is great. However, there are some limitations. It's developed in Python 2.7, which make it really hard to rerun and retrain. And it depends on the old version of Keras and TensorFlow. Most important of all, it was trained on the old version of Gencode annotation. Therefore, I developed Splice AI Toolkit. It is an easy to retrain and rerun framework in modern PyTorch framework. And now since we have MEN annotation, which was published in Nature back in 2022, MEN is the annotation that takes the consensus between NCBI RefSeq and Gencode annotations. And we retrain on MEN and also RefSeq annotation. Here's the result. We calculate the AOPRC, a top K accuracy, AUPRC, precision, recall, and also F1 score. We can focus on the precision. So the orange curve is the newly trained Splice AI model using Splice AI Toolkit, and the blue curve is original Splice AI model. We can see that our model is more precise, and we also observe that when applying original Splice AI model to the MEN dataset, the precision drop, which imply overfitting. And furthermore, we apply Splice AI Toolkit to retrain Splice AI for different species, mouse, zebrafish, Arabidopsis thaliana, and honeybee. And for mouse, we can first focus on mouse. We can see that, okay, so the green line is the original, it's the new model that we trained, and the blue curve is the original Splice AI model. We can see that the recall remains, however, precision drops for the original Splice AI model. And when it comes to more distant species, both precision and recall drops. And the results show that we need to retrain Splice AI model on specific species that we're interested in. Okay, so this is how we use Splice AI. And this is the concept that people can further use splice AI to predict original sequence and introduce the mutation, and they can predict the cryptic new splice site. And another concept that Splice AI Toolkit introduced is called model calibration. So you get a score from Splice AI. We have the question that what does Splice AI score signify? And do models' prediction probability actually represent the true likelihood of an event happening? So the concept is called model calibration. This is a bit branch out from the main topic I'm talking about, but I think it's an important concept. And if you're doing any modeling, it can be easily applied to your research. So I want to spend some time talking about model calibration. And I will use a simple binary Chihuahua classifier as an example. So imagine you train a model that can classify if a given image is Chihuahua. And this image is a Chihuahua on the grass, so it's a perfect Chihuahua, so the model gives it a very confident score. And in this image, Chihuahua is driving, so probably the model is less confident and give a less confident score. And in this one, it's sipping juice on a chair, beach chair, so probably the model gives a low score. And this is a headshot of a Chihuahua. Oh wait, I got confused, and model also got confused, so it gives a relatively higher score. So now we input all the images into the model and put them into 6 bins of scores. And for each bin, we calculate its empirical probability. So that's focused on the first bin. This is a golden retriever, so the empirical probability that the model predict correct for this bin is 0.5. And we calculate this empirical probability for each bins that we binned. So this plot is called reliability diagram, and the blue dotted line is perfectly calibrated. So now what we want to do is have a universal nonlinear scaling that scales the model's output scores without changing the model performance. And in this case, the pink curve is before calibration and the red curve is after calibration. So how do we do it? A common approach includes isotonic regression, histogram binning, and also Platt scaling. And the approach that we adopted here is the single parameter variance of Platt scaling, which is known as temperature scaling. This is very widely used in deep learning. So after the model output the logit, instead of directly input into the softmax layer, we can scale it by the temperature parameter and then fit into the softmax. And if you're interested, you can look into this paper. It discusses more about model calibration. So here's the result after we calibrate the Splice AI's model's prediction. I will just quickly talk about the 2 loss functions that people widely use, which is negative log likelihood and expected calibration error. And these assessments show that after model calibration, the model output probability actually better reflects the empirical probability. So in summary, Splice AI Toolkit takes the genome and annotation and modularize every step to help the users to do data preprocessing, to model training, and fine-tuning your model. We also pre-trained Splice AI on human man and also different species, and it can be used to predict genetic variance effect on splice site. Furthermore, we introduced a new concept called model calibration to make it a more useful tool. So problem solved? Not really. So one of the biggest concerns that we have about Splice AI is its canonical transcript labeling approach. It only selects one isoform for labeling. So here's an example. This is a gene locus, and for this gene locus, it produces 3 isoforms. And that's focused on the second isoform. We can see that the red axon was spliced out. So actually, there's a splice junction here. However, from the experiment that we conducted, we observed that Splice AI gives a very low score for alternative splice sites. And if you're using Splice AI to analyze your splice junctions, you will miss a lot of alternative splice junctions. Therefore, I developed SPLAN. And the main novel idea that we introduced is to focus on the splice junction level. And we want the model to learn the local regions around each splice junction. So instead of using the 10K flanking sequences and capture the whole region, we use the local region 400 base pair around donor and 400 base pair around the acceptor. So we concatenate them into as our model input. And this is the model architecture of SPLAN. And also it's a good example to introduce how people usually build their convolutional neural networks in a modern deep learning model. So the input first goes through a convolution layer, and then it goes through four residual blocks, with each residual block composed of a conv layer, a batch normalization layer, and ReLU, and repeat twice and a residual connection. Now, four residual blocks form a residual group, and between two residual groups, there is also residual connection. We repeat it 5 times, and finally, we output into a conv layer, flattened layer, dropout layer, a fully connected layer, and softmax layer for the final probability output. So for Splan, we show that the score-wise is more stable compared to Splice AI. And also, we simply use the Splan score to remove some spurious spliced alignment, and actually it can improve transcriptome assembly. And people might be curious about interpretability of Splan. So we conducted a ablation study, which is removing certain layers in the neural networks and observe its contribution to the full model performance. We also changed the input sequence. Right now it's 800. We try different flanking sequences to understand what's the amount of information that's needed to let the model learn the splicing pattern. So in summary, if you want to have a reliable splice junction scoring system, I would suggest you trying Splan instead of Splice AI because it learns the splice junction signals. And also we show in the paper that Splan also generalized to non-human species, including distant ones like Arabidopsis thaliana. And I assume people might build their own deep neural network, so I'll share some technical takeaways. So instead of using the classical cross entropy, I use focal loss. So basically, you can see the formulas here. They're the same. Instead, it's just focal loss. It has another term. So concept-wise, it's simply just focusing on those harder to predict examples and penalize them more. Therefore, the model learns faster and also do better on predicting those hard to predict examples. And we use adaptive learning rate with warm-up and sinusoidal decay. And you can try residual connections. And also there's a group parameters in the convolution node layer. Okay, so what is the future of sequence models in genomics? I want to spend the last 2 minutes quickly talking about my perspective. So most people think it is transformer. So first I'll talk about its application to protein sequences and also the DNA sequences. This is a timeline of the protein transformer-based models that I summarized. People can combine the core component of transformer, the multi-head attention layer, with the CNN layers to train a model from scratch for a specific task. Or recently, there has been significant interest in a paradigm of foundation models. So this term is used to describe the machine learning models that initially use a self-supervised autoregressive approach to pre-train on a large amount of generalized unlabeled data. And then we can fine-tune the foundation models to predict a wide range of specific tasks. And in the application to protein sequences, it is very successful. Famous ones include Rosetta Fold from University of Washington, and OmegaFold from Helixton, ESM-Fold from Meta, and of course, AlphaFold from DeepMind. And in my opinion, it is relatively mature. However, in contrast, transformer-based DNA sequence models are not yet mature in my opinion. Examples include Informer, DNA Bird, AS4 state models, Hyena DNA, along with more recent ones like Nucleotide Transformer by InstaDeep, and Evo, which was developed on the special model architecture that they proposed, Striped Hyena, and was released by Stanford and ARC Institute. So challenges remains, particularly with the substantially longer DNA sequences, and it's harder to capture the long-range interactions and dependencies. Additionally, with the current limit up to around 100,000 base pairs. So protein-wise, we found a lot of great applications such as protein folding and synthetic protein generation. What about DNA? The current CNN models has been applied to predict epigenetic marks, 3D organization, and gene expressions, and of course, splice site prediction. And I believe transformer-based model can do better, and you can look into this review paper, which I found it really helpful. And also there are some attempts to apply deep learning sequence models to predict genome annotation, but in my opinion, it is very preliminary. And just quickly advertise my tool. Recently, I released a new tool called Lifton. It maps the annotations from the reference to the target, and it can be applied to map from, for example, GRC38 to CHN13 and improve the protein genes. So I want to thank my PhD advisors and also all the collaborators, all friends at Malone and CCP, and all Hopkins friends. And last, I want to end my talk with one of my favorite quotes from Demis Hassabis. So if you think of mathematics as the perfect description language for physics, then AI might be the perfect one for biology. And with that, I'm happy to take any questions.